I'm Heidi Basler, and I'm a lecturer affiliated with the Hull York Medical School, but based here in psychology. In my lab, we work with people that are partially blind or completely blind or else deaf, and we're interested in the way that their brain compensates or adapts to that kind of loss.
So I'm doing a master's project at the moment, and I'm looking at structural images of people's brains. So we're looking at developing a scan that will allow us to assess the amount of myelin in someone's brain at two time points and also between populations, so people with MS, and compare it to normal brains, for example.
Alex's project was about helping us to develop a technique in the MRI scanner that allows us to look at a substance called myelin in the brain. Now, myelin is a substance that insulates nerve cells and allows them to conduct electrical signals more efficiently. And when people have diseases like multiple sclerosis or MS, myelin gets attacked and degrades so that the nerve cells can no longer transmit information anymore. And so we were interested in being able to measure the changes in myelin levels in patients.
So one of the things that we're really interested in using this for in the future is monitoring and diagnosing disease progression. So if you can have a scan that is higher resolution and shorter than classic clinical scans, you can detect differences in people's brains at an earlier date, and this allows you to test whether a drug is working or test whether you need to go down a different route, but also is important for that initial stage of diagnosis of, is this one thing, could this be MS, could this be something else? So this is important for deciding what course of action to take in clinical trials and things like that.
So MRI scanners are really good at taking pictures of the brain and showing us the shape and structure of different parts of the brain, but they're not very good at quantifying or measuring how much of different substances are present in the brain, such as myelin. So we wanted to develop a technique that allows us to do that. Students get the opportunity to work in the Neuroimaging Center and actually not only participate as subjects, but also collect the data. So they're there in the Neuroimaging Center using the equipment and analyzing the data firsthand. Students have to be involved in all aspects of the project. So designing the project, coming up with a protocol, recruiting subjects, running subjects in the neuroimaging equipment, analyzing their data, and even publishing papers.